Hello, everyone. This is Paul Orchard. Welcome to the MLD Standard of Care meeting sponsored by the Cure MLD group. We have a wonderful presentation this morning. This is the third of the series. The first two series discussed the diagnosis and the disease and the role of newborn screening. Today, we're going to focus on therapies for MLD. And we have a series of international speakers here that are well known in the field, and we have a great opportunity to both hear what their experiences are and where they're going in terms of therapy, as well as to have an opportunity to have a panel discussion afterwards. The Cure MLD is an international organization, advocacy organization. It's been important in supporting families that are touched by MLD. They've also provided information to families and providers as well in discussions such as the one we're having today. We're going to talk about several different things in terms of our presentations today. Gene therapy, including ex vivo gene therapy as well as in vivo gene therapy, will be some of the topics. We're also going to talk about the role of enzyme therapy and the relative risks and benefits of all these things. So my name is Paul Orchard, as I mentioned. So I'm a pediatrician trained in hematology, oncology, blood and marrow transplantation. We've been interested in diseases such as MLD for extensive period of time. We've had about 30 plus years of experience in transplantation for these diseases, but it's very clear that we need new opportunities to treat these patients. And again, we're excited to talk about some of those today. So one of the important things to remember, so we're going to have a series of questions that are already been previously prepared, but for those of you that are listening and have questions, we're not going to have a question and answer period with each presentation. We're going to save those to the end. So if you would like to put in your question in the question and answer tab below, we'll get to as many questions as we can. So I'd like to move ahead in introducing today's speakers. So the first speaker we're going to have today is Dr. Gaspar, who is one of the leaders in Orchard Therapeutics and has developed, their group has developed lentiviral-based therapy for MLD, and it has pretty extensive experience in Europe. So Bobby's going to talk to us about that. Dr. Whiteman in Takeda has a long history in genetics and metabolic disorders, was at the Mayo Clinic for a period of time. He's going to talk about enzyme-related therapies. And Dr. Seymour at Homology also has extensive experience in gene therapy, and we'll talk about the in vivo approaches there. And Dr. Dietz is someone we've added here just in the last week or so, and he works at Bluebird Bio and is a leader in some of the gene therapy programs there. And we'll talk about some of their experiences in gene therapy for other leukodystrophies and other disorders. So having said all that, we're going to have 10-minute presentations plus or minus from each speaker. And then, as I mentioned, we'll have a series of moderated questions. And then we'll get to questions and answers that the audience has towards the end. And we'll finish up at noon Eastern time. So again, thanks for joining us today. And I will turn this over to Bobby.
Thanks, Paul, and thank you for the invitation to talk, and thank you to Cure MLD for inviting me. And so, as Paul says, I'm Bobby Gaspar. I'm CEO of Orchard Therapeutics. I'm an academic physician by training and have been in the field of hematopoietic stem cell gene therapy, which is what I'll talk about today, for over 25 years, and have worked on the laboratory-based mechanisms of this approach, and also in the clinical trials of this form of gene therapy, both for immune deficiencies and neurometabolic disorders as well. So it's a real pleasure to be able to talk to you about the approach that we're taking for MLD and other neurodegenerative neurometabolic conditions. So if we go to the next slide, please. So Orchard came into being from a history of academic development in the field of hematopoietic stem cell gene therapy, and having seen some of the benefits of this approach in immune deficiencies and certain neurometabolic conditions, really Orchard was formed in order to try and take these developments to become approved genetic medicines for patients with these very severe conditions, and to establish ourselves really as a global leader in hematopoietic stem cell gene therapy. The programs at Orchard that are in the clinical setting at the moment are focused on rare inherited conditions where there are no treatment options, or where current standards of care are limited or have unacceptable levels of morbidity and mortality. And the company is dedicated to transforming the lives of patients with these severe diseases. More recently, given what we've seen in rare diseases, we also think this approach can be used in larger indications as well in the future. So next slide, please. So what I want to do really in this short presentation is just talk about how this approach of hematopoietic stem cell gene therapy, or HSC gene therapy, can work to meet the needs in these severe neurodegenerative conditions. And I suppose it's really about saying, well, how can you use hematopoietic stem cells to try and prevent neurodegeneration in the CNS? How does that work? And that's what I want to spend the time doing in this short presentation. So if we go to the next slide. And this is really the crux of the approach. What we're doing is we're taking the patient's own blood stem cells, or hematopoietic stem cells, and introducing into those cells a working copy of the gene that is missing. Now, the way we're introducing the gene into those cells is using lentiviral vectors. And so these vectors carry the gene, the therapeutic gene, into the patient's hematopoietic stem cells. And what lentiviral vectors do is actually they integrate that therapeutic gene into the genome of that target cell. So essentially, the gene gets hardwired into the DNA of the target cell. And what that means is, as that cell divides, that genetic information is passed on. And so through that process of integration and the passing on of genetic information with each cell division, through also the fact that hematopoietic stem cells have this ability to self-renew, so they make new copies of themselves. And as I say, by making new copies of themselves, we're passing the genetic information onto those new copies. We have the potential, through a once-off administration, to have a long-term and durable outcome. So I think that's one very important point, that a single administration of these gene-modified cells can have a very long-term, durable effect. The other very important thing to say, and this now comes to how we think these, the modification of these cells can correct or potentially address neurodegenerative conditions, is because these hematopoietic stem cells have the ability to become multiple different cells in the bloodstream. So they can become immune cells, which is why we have used HSC gene therapy to address certain immune deficiencies. They can become red blood cells, which is why it's being used in hemoglobinopathies. But also, these cells have the potential to become cells called monocytes or macrophages, and they can cross the blood-brain barrier. So they have a natural ability to cross the blood-brain barrier and become cells called microglia in the brain. And so by delivering a gene to hematopoietic stem cells, and through their ability to cross the blood-brain barrier and get into the brain, we can deliver therapeutic genes to the CNS. And that's the mechanism by which we hope that this approach can prevent neurodegeneration in these devastating disorders. And I'll talk a little bit more about that in just a bit later, but the next slide, if you could go to the next slide, please. This is what patients will go through in order to receive this type of gene therapy. First, we take these cells, take cells from the patient themselves, and we can either do that directly from the bone marrow, or increasingly we do that by moving the cells into the bloodstream and taking it from the bloodstream. We purify the stem cell population. So this is a proportion of all of those cells. So we purify that stem cell population. And as I said, we introduce a working copy of the gene into those cells using a lentiviral vector. These cells can then be stored down, cryopreserved, where we can do quality control to make sure we've got enough cells, that they're viable, that there's enough gene in those cells. And then these cells are shipped to the patient center. The patient receives conditioning to make space for the incoming cells, and then the cells are infused into the bloodstream of the patient. And so if we then go to the next slide, this is just telling you how those cells that we introduce into the bloodstream can then go and affect neurological conditions. These gene-modified hematopoietic stem cells are circulating in the bloodstream. As I say, they have this ability to cross the blood-brain barrier. And when they cross the blood-brain barrier, they can distribute into all of the different sections of the brain. They become these specialized cells called microglia. And because they've been gene modified, they secrete that therapeutic gene, that therapeutic enzyme or protein, which then can be taken up by neurons. And those neurons, which previously did not have the enzyme, now have a working copy of that gene, of that enzyme, and they're able to prevent the buildup of substrates that cause neuronal death. So now these neuronal cells have the therapeutic enzyme and are able to resist that buildup of substrates. And so that's the mechanism by which we believe we can prevent neurodegeneration in these devastating conditions. So I think this is a very exciting approach. And so look forward to addressing questions later on. So thank you very much, Paul.
Bobby, appreciate that and giving the overview in terms of the platform that lentiviral therapy allows us. I think maybe later on we'll discuss a little bit more about how that approach differs from allotransplant with the experience there, but we'll get to some of those discussions later. So the next presenter is Dr. Whiteman, who, as I mentioned, is taking a leading role at Takeda, genetics and metabolism, and is going to talk to us a bit about Takeda's experience in terms of opportunities to treat MLD. Dr. Whiteman?
Thank you, Paul. So what is now the Rare Genetics and Hematology Unit within Takeda has a long history going back into the 1980s, the 1990s, when a number of companies, Genzyme, Biomarin, Amgen, others, began to work on replacing enzymes as a treatment for various genetic metabolic diseases. And that was an era when the genes were being finally identified. Technologies to allow gene expression in different cell types are being developed. The possibility to scale this up and commercialize it in a way that could be viable both from a clinical, medical, and a commercial perspective was developing. And TKT, which was one of those companies that started, initially worked on treatment of Fabry syndrome and Hunter syndrome. And we then began to consider what other diseases we might tackle with this approach. Now, in contrast to the gene therapy you've just heard about from Dr. Gaspar, this is really older technology biochemically. The ability to do what we're now doing in the laboratory really has been feasible for almost 40 years at this point. However, we continue to pursue it for selected diseases because it provides, as it were, an alternative approach, and because, as we'll probably end up discussing later on, it may address the needs of some patients, particularly in MLD, who unfortunately will not necessarily be able to access the gene therapy. The concept is simple. You take the gene, you set up an expression system in an animal cell line, and you produce the protein. And then you administer the protein through any of a number of routes. The execution is much more complicated. You have to decide on the appropriate route for the target of the disease. you have to deal with the possibility of the development of antibodies, both in terms of the generation of infusion reactions, most of which are manageable, but some of which can be severe. And you have to assess whether the impact of those antibodies on efficacy, i.e., does the development of antibodies stop the therapy? Does it weaken its ability? Does it kick in later on and prevent what was initial response? In terms of specific enzyme therapy for MLD, both TKT and a company in Denmark started working on this around the same time, but with different approaches. The group in Denmark had based their project on some animal work that had suggested to them that intravenously administered enzyme might reach into the central nervous system because the blood-brain barrier in the mice that they had been studying is damaged as a consequence of the disease process. And they took that as far as a phase 1 human clinical trial in a small number of patients, and had results that initially seemed promising. At that point, TKT had become Shire or part of Shire Pharmaceuticals, and they acquired that program and brought it in-house and ran a replicating trial. An important part of any scientific evaluation is replicating one's results. Unfortunately, that trial showed that intravenously administered enzyme was not effective, either in terms of the biochemical assessment of it reaching the various compartments of the brain, or unfortunately in terms of clinical response. And at that point, that line of development was terminated. The Shire decided to proceed with an approach to intrathecal administration, in other words, directly injecting the enzyme into the cerebrospinal fluid, bypassing the blood-brain barrier. And large molecules like enzymes cannot by themselves cross the blood-brain barrier. We don't have any representative of any of the organizations that are doing this here to discuss today, but there are a number of smaller companies that are attempting to develop what are broadly referred to as Trojan horse molecules, which are coupling of enzymes to other molecules that can cross the blood-brain barrier. And in very early development, some people are looking at that. That would allow an intravenous therapy that could deliver the enzyme centrally, although there are many hurdles to overcome before that can be achieved. So we have used an intrathecal drug delivery device, or an IDDD, and that was largely developed for this purpose, for this disease and some other related neurodegenerative diseases that we work on by a partner company. We embarked on a phase 1 clinical trial, a dose exploration and safety trial, back in the late part of the first part of the decade, early second decade. And that involved a small number of patients, primarily in Europe. And the trial as a whole was generally not viewed as particularly successful. Unfortunately, all the patients who were enrolled in that trial ultimately progressed to the most severe level of MLD. However, we did observe some dose response effects in looking both at 2 individual patients and at some of the group data from the trial. Specifically, there were 2 patients in that trial who were younger siblings, one of another patient in the trial and one of a patient who had been in the original Denmark trial. And so we knew their clinical course, and we were able to demonstrate that the 2 younger patients' progression of disease, whilst inevitable, was delayed by about 2 years. In conjunction with that, we found various biomarker changes, dose-dependent changes in the cerebrospinal fluid concentration of sulfatide, dose-responsive changes in the appearance of the and ratios of volumes of the MRI, and most importantly, perhaps, in the ratio of something called NAA to creatine, judged from a MR spectroscopy analysis, a specialized form of MRI imaging that allows you to semi-quantitatively estimate the concentration of compounds in the brain. NAA is the principal amino acid in our neurons normally, and it's a good marker judged against the ratio of something called creatine, which is fairly stable, of the preservation of neurons. So if your NAA creatine ratio is higher, then you are preserving your neuronal cells. On that basis, we decided to move forward with a phase 2 trial to explore, again, in a relatively small group of patients, further the dose response and also the potential response of patients whose disease was more advanced. And this occurred at a time when I think a lot of people were hoping that the original Milan gene therapy would literally become the cure for the disease. I'll let Dr. Orchard discuss that later on. But clearly from the original data, it appeared that patients with more severe disease were not responding as well as those who were pre-symptomatic. And we experienced a large increase in demand for access to the enzyme that we've been developing, as many of you are aware. So we organized the present clinical trial, a phase 2b trial, and designed it in such a way that we could look at various degrees of progression. There is a primary efficacy group in the trial who are patients very similar to those who've been enrolled in the Orchard trials with early stage disease, and that is powered statistically to assess the overall efficacy of the compound. And then we have several subgroups within the trial to allow us to look at patients who were, as it were, starting from a later point in the disease and ask the question, can we at least stabilize them and stop them getting worse? The trial was designed with a long treatment period of 2 years. We decided based on the dosing data from the phase 1 study that we needed to substantially increase the amount of enzyme, the amount of protein we were placing in the CSF. So we went to a much higher dose given weekly. And we've just completed enrollment of the trial last week. The treatment period runs for 2 years. So the analysis of data from the trial will not begin until 2 years from now when the last patient leaves the trial. And the assessment is based on two things: the clinical assessment of the GMFC MLD score developed by the group in Tübingen in Germany originally, and video-based assessment of the motor function of the children using the same scoring system by a group of independent observers. And I think I'll stop at that point and address any questions that may come up later on. Thank you.
Thanks, Dr. Whiteman. I appreciate your giving us an idea of what kind of opportunities that enzyme therapy might have for MLD and what some of the limitations might be there as well. So yeah, it will be important to talk later about what patients might be eligible for those types of trials down the road. And I'd like to learn more about some of the outcome pieces too, but we can get to those things a bit later. So I have the opportunity now to introduce Dr. Albert Seymour, who's the Chief Scientific Officer at Homology. Homology is also interested in gene therapy approaches. We were talking about before with Dr. Gaspar, the lentiviral-based approaches, which are essentially taking cells, the blood stem cells out of the body, engineering them to make enzyme and replacing them. So that's what we would term an ex vivo approach, meaning outside the body. But there are opportunities for gene therapy in terms of in vivo approaches too. And I think Dr. Seymour will talk to us about opportunities there, because some of these have developed into clinical trials and are being used in diseases like PKU. So Dr. Seymour, thanks for being with us today and sharing what you're doing.
Yeah, thank you, Dr. Orchard, and thank you to the Cure MLD for giving us the opportunity to present on behalf of Homology Medicines and the work that we're doing here. If you go to the next slide, Laura. I do have to highlight the forward-looking statements. We're a publicly traded company, so some of the material that I can present can be deemed as forward-looking. So any questions on that, feel free to go to our SEC filings. Next slide, Laura. I'd like to start with this in terms of, I think, what you heard a little bit earlier from Dr. Gaspar, as well as really the key mission at Homology is we feel that we're at this precipice of having technologies to allow us to evaluate the potential to not only treat disease, but perhaps cure genetic disease. It's something that really drives us at Homology to try to utilize these technologies to really get at the underlying cause of many of these rare genetic diseases, including MLD. Next slide, Laura. The underlying foundation of Homology are these 15 AAV vehicles that were originally discovered in human hematopoietic stem cells. So we call them AAV HSCs. That's where that term comes from. And if you look at the top part of this slide, you'll see this sort of purple figure. That is a representation of the capsid itself. So we think about these AAVs as basically drug delivery vehicles. And so we can utilize and take advantage of the AAV that will bind to a cell and then enter that cell and then go into the nucleus of that cell. And then once it gets into that nucleus, it'll deposit and release the vector genome. And so we can engineer the inside of these AAVs to take advantage of the viral capsid to deliver this vector genome into that cell. And from a gene therapy perspective, what we can do in engineering that is we can engineer so that we put in what's called a promoter. In the case of MLD, we're using a promoter that would be expressed in any cell that it enters into. And then directly next to that promoter, we put in a fully functional corrective gene. So in this case, it would be aryl sulfatase A, ARSA, the full cDNA. And then what happens when that AAV enters the cell, gets into the nucleus, it forms what are called episomes. And these are the circular forms of this vector genome. And then the cell's machinery will recognize that promoter sequence, that green sequence, and it will turn on the template making mRNA to ultimately make the protein for aryl sulfatase. And this is sort of the approach that we utilize in using these AAVs as delivery vehicles. Next slide, Laura, please. One of the things that we did as a company very early on is we had these 15 AAV HSCs. We wanted to see if we do an intravenous, an IV, directly into the vein administration, where do they go? We had to characterize all the different tissues that these AAVs may go. And we felt the best place to test this would be in non-human primates. And so we actually published a couple of years ago some of the biodistribution data on these particular AAVs. And one of the things we found that after an IV administration, we do get broad distribution across many different tissues. And one of the things that got us very interested in developing these for CNS type related diseases is the fact that they do cross the blood-brain barrier, meaning we can inject into the IV, into the veins, and we can hit the liver, we can hit the lung, we can hit the muscle, we can hit different tissues, but we also get into the brain. What you see on the bottom part of this slide is that if you look in the brain, you can see anywhere that's stained brown is evidence that we've crossed and we're expressing a protein in those particular cells. And so we see it in different regions, sort of broad distribution throughout the brain. And then when we break it down and look at different cells, depending on the region of the brain that we're looking at, is in the deep brain tissue, you can see we are targeting neurons, but we're also targeting cells of glial nature. And most of these are predominantly astrocytes, which is a cell type that's very good at secreting a particular protein. But then when we look in some of the peripheral nerves, you can see here we're hitting predominantly neurons as well as some of the glial origins. And what you can see on the right is just some further high-level imaging or high-focus imaging in different regions of the brain, looking at and showing, so anywhere there's dark or the green is areas that we're seeing expression on that side of the brain. So taking that into account, we felt that we had the drug delivery approach with these AAVs to target the right cells for MLD. Next slide, please, Laura. This is how we came up with HMI 202. This is our gene therapy development candidate for MLD. And basically, if you look at this middle part here, that's kind of what the viral capsid looks like. It's this protein encapsulated. And then on the inside of that, again, we have a promoter, and we call it a ubiquitous promoter, meaning that we want it to be seen and recognized by whatever cell type that it goes into to turn on human aryl sulfatase A. And so we put that right next to that promoter within this vector genome. We can then start to test in preclinical models how that vector works in terms of delivering this gene sequence to the different tissue, the relevant tissues, both peripherally as well as in the CNS for MLD. And so I'm going to walk you through some of the data that we've generated using this approach in the MLD mouse model. Next slide, please, Laura. The first thing that we had to assess is whether or not we truly cross the blood-brain barrier, meaning if I inject it into a vein, do we not only target the peripheral tissues, do we get across into the relevant tissues in the brain? And we wanted to make sure that we could do that both in the mouse model, but also in non-human primates with the HMI-202. So anywhere you see staining that's brown, these dark browns in the mouse, you can see that we are targeting the dorsal root ganglion, which is part of the peripheral nerves, motor neurons, motor cortex, as well as in the cerebellum in the brain. We then, in order to evaluate this in the non-human primates, we actually had to put a tag on the protein because the non-human primates make an aryl sulfatase A that we need to be able to distinguish the human one from the mouse one. And so we put a tag on there. And so here, if you look in the purple, you can see that that's evidence that we're making aryl sulfatase A in this case, the dorsal root ganglion. You can see purple staining here in the motor neurons, and we also counterstain with lamps you can see in the lysosome. Here we see it in the motor cortex, and then ultimately we see it in the cerebellum. So we felt confident that with this IV dose, we could cross into the relevant tissues. Next slide, please, Laura. Then what we wanted to do is make sure that we were able, after a single administration, that we could see activity in the brain tissue. Now this is in the mouse model, and we wanted to make sure that that was sustained over time. And so what I'm showing here is after a single administration, IV, as early as 1 week, we start seeing activity levels in the brain of these mice. And then that goes and is continued out through a year, so where we have the data on this. So we felt now very confident that we can inject into the vein, we get across the blood-brain barrier, and we're seeing evidence of activity of aryl sulfatase A in the right tissue all the way out to 52 weeks. So with that, the next study we did, next slide, please, Laura. We then wanted to make sure that now that we see this activity, do we see an effect on certain biomarkers? And so the one we wanted to look at were brain sulfatide levels. And so sulfatide obviously is the substrate that will accumulate in the absence of aryl sulfatase A in the lysosomes. And so what you can see here is over time, these are, I have this listed as normal. These are the aryl sulfatase A mouse model that did not receive HMI-202. And you can see that we have elevations about 10 times more than wild type controls or normal controls of sulfatide. In the treated animals, we virtually normalized that. So we have the sulfatide levels that are down to the same level as a mouse that does not contain a mutation or an absence of aryl sulfatase activity. And this was done at 7 months after we dosed these animals. So we felt very confident that we could get there, and then it was active as it relates to sulfatide reduction. Next slide, please, Laura. We then wanted to look, do we have evidence that we improve the health of the lysosomes? And so here we use a marker called LAMP1. And what you can see here is if you see really dark staining, that would be evidence of LAMP1 accumulation in the lysosomes. This is a hallmark of lysosomal health. And so the more LAMP1 accumulation you have, the less healthy that cell or that lysosome is. And so what you can see here in the untreated, or in here in the slide we call it a normal untreated, you see a lot of this dark staining. But in the HMI-202 treated, you see a significant reduction in that amount of staining. So we see a LAMP1 reduction in these animals at 3 months post-dose. And this is looking directly in the spinal cord tissue of these particular mice. So then we felt very confident that we're getting to the right areas. We're having an effect of the drug. It's active, it's reducing sulfatides, it's improving the health of the lysosome. The next thing we wanted to look at is, do we see a phenotype in the mouse? And so if you go to the next slide, please, Laura. One of the things we did was we took these mice and we treated them, and then we aged them over time. And so you can see here the age that we treated them. And so in this case, they were about 2 months old when we treated them, and then we followed them for a very long time. And we put them on what's called a Roto-Rod assay. And basically what this does is this will allow us to assess whether or not our treatment has any effect on a motor benefit. And so what you look at is these mice run on a rod, and then you measure how long they can stay on there as you increase the speed of this rod. And what you'll see is as we get out to maybe 7 to 9 months post-dose, where the animals are around 9 to 11 months old, we see a significant benefit of the treated mouse over the untreated, meaning that the untreated start to fall off the Roto-Rod much sooner than the treated mouse. So this also then gave us evidence that we have a phenotypic effect in the ARSA knockout mouse model. Next slide, please, Laura. So that really brings me to a summary of where we are. This is a lot of the preclinical data that we've generated on this. We've achieved high levels of human ARSA activity in the relevant tissues, particularly the CNS. We do see it in the peripheral tissues as well. We have regulated disease-relevant biomarkers in the CNS, primarily the sulfatides, as well as a LAMP1 reduction. And by detecting it in the relevant regions in both mice and non-human primates, we felt that was very important as we think about pursuing and progressing this out of the preclinical research arena ultimately into the clinic. And so where we are as a company, we've taken a lot of these learnings from the IND-enabling studies, and what we're doing is we're further optimizing HMI 202 before we initiate that clinical trial. And what I mean by that further optimizations, we're actually evaluating the expression levels both peripherally and making sure that we can maintain that in the CNS, the relevant areas. And so if you go to the next slide, Laura, I'll just end here and just really say that we're committed to supporting MLD and rare disease communities. And on the left, you can see a picture of the whole Homology team outside our headquarters in Bedford, Mass. In the middle, you can see there's Maria. So when she came to visit, I think everyone really enjoys having the opportunity to meet with patient advocates and the rare disease community. And on the right are some of the scientists that are actually working on the MLD program. So I'll end there, and obviously happy to take questions at the end.
Great, Dr. Seymour. I appreciate the opportunity to have you talk to us about the experience you've got both in the animals, the mice and the primates. So that's very exciting data thus far. The final person who's going to be presenting today was added on just in the last week or so. We appreciate his flexibility in helping us or helping be with us today. So Dr. Drew Dietz is trained in blood and marrow transplant and is now Bluebird Bio and is the Chief Medical Officer there in leukodystrophy trials for adrenal leukodystrophy, which is related leukodystrophy. And he's going to spend a little bit of time today talking to us about what Bluebird Bio does and their experience. So thanks, Drew, for being here and helping us out.
Thank you, Dr. Orchard, and thank you to the Cure MLD for inviting me to participate today. Thanks for the slight upgrade there. Definitely not the Chief Medical Officer, but certainly do oversee our cerebral adrenal leukodystrophy trials within Bluebird Bio. So just to give a brief overview, you heard an excellent presentation from Dr. Gaspar about ex vivo lentiviral gene therapy, and Bluebird Bio is a gene therapy company as well, with a focus on rare diseases. And there's a couple areas in which we use gene therapy, and in particular lentiviral vector therapy. One is to actually use that lentiviral vector to transfect T cells and change the immune system to try and fight cancer. So we have open clinical trials in multiple myeloma. The other area of focus in the company from a rare disease standpoint is in using that lentiviral vector to transfect hematopoietic stem cells, and through a very similar process that Dr. Gaspar described, be able to deliver those gene modified cells back to the patients with those rare diseases. There's three areas where we have studies that we've been conducting. One is in sickle cell anemia, one is in thalassemia. So two of the areas of hemoglobinopathies or blood disorders. And then we also have a program, as I mentioned, that I oversee with cerebral adrenal leukodystrophy. So another neurometabolic disease that can affect the brain, particularly of young boys, as it's an X-linked disease. with a similar goal of trying to replace back the defective gene to be able to produce the protein that is otherwise absent or abnormal for those patients, and hopefully see beneficial treatment effect. So that's kind of the overall overview of Bluebird Bio. As Dr. Orchard mentioned, I'm a pediatric hematology, oncology, and stem cell transplant physician myself. And I've now been with Bluebird for about the last 3.5 years. And for the last couple of years have been overseeing the clinical research and development program for our rare cerebral adrenal leukodystrophy ex vivo gene therapy.
Great, thanks, Drew. We appreciate your being here with us today. So again, what we're going to move to now is a panel discussion. We've got a number of questions that I'd like to ask the panelists, and but if there's people in the audience that have questions that have come up through the presentations or through the discussion that we're going to have, if you'd like to pose those questions in the Q&A tab, we will get to as many of those as we can. I'd like to start out by asking Dr. Whiteman a bit. So one of the issues in clinical trials for rare diseases like this is that how does one show that you bent the curve in terms of the disease outcomes? And there are clinical disease outcomes that end up being clearly important in terms of cognition and motor related problems, as you touched on. But are there things that we can do beyond that will give us a sense as to how well our interventions are actually working, and potentially things that will be meaningful for the agencies such as the FDA in the US and the EMA in Europe? So, and Dr. Whiteman, you talked a little bit about biochemical markers that may be important, and also a bit about imaging and new opportunities that might be available there. Can you expand on those a bit for us?
Sure. And, I think particularly when one's working with a rare disease, it's important to try and build a picture of efficacy, which is a word we've all heard a lot about recently because of the COVID vaccines, from multiple sources. There's a huge difference between trying to do clinical development and clinical trials to the satisfaction of regulators for common diseases like asthma and diabetes, where you can round up 10, 20, sometimes 50,000 patients and study them and answer a lot of questions in a mathematical, statistical way. In our area, one has to attempt to do that, but one has to bolster that with other lines of evidence. So I really look at 5 lines of evidence of trying to demonstrate efficacy. The first, as you mentioned, is biochemistry. These are at the most fundamental level, genetic errors in metabolism and biochemistry. And so one ought to be able to show that one can deliver one's enzyme, as several of the speakers have, to the appropriate location, either through a virus gene therapy or deliberately. One ought to be able to show that in doing that, you're delivering a high enough concentration of the enzyme to plausibly correct the deficiency that the patient has. Then one wants to show that the accumulating substrate, in this case, the sulfatides, the toxic metabolites, are reduced as a result of the presence of that enzyme. And there one needs reference data sets of levels of the metabolite in untreated patients as well. And then thirdly, one wants to look at other biochemical markers, in this case, particularly those of neurodegeneration. So in the cerebrospinal fluid of these patients, we're examining the NAA creatine ratio, both from the MRS perspective and actually quantitatively in the fluid. We're also looking at common markers of neurodegeneration, many of which have been developed for studying adult diseases like neurofilament light and tau, and essentially assessing secondary biochemical indicators of the level of damage or otherwise that's occurring in the central nervous system. Then one wants to look at what people would define as more traditional clinical outcomes. These would include physiological measures, such for some diseases, one will be looking at directed heart function, pulmonary function, liver size, spleen size, not particularly relevant here. In this case, we're looking at motor function. And so we're actually using in parallel two instruments of motor function. One is the GMFC-MLD Clinician's Rating Scale, which is essentially, I think many of you are probably aware of it, but it's essentially a 7-point scale that runs from 0 to 6 that indicates the level of motor functioning of a patient with MLD. And then the GMFM-88, the Gross Motor Functional Motor Scale, with an 88-point questionnaire. It's actually not just a questionnaire. Some of the items require the child or the parent to demonstrate certain things. We've had a lot of discussion, which I'm happy to share with those of you who might be interested in it, particularly in the other companies with the FDA, about which of those is the best way to approach this. Currently, we're focused on the GMFC-MLD. However, we're gathering data from both instruments. And then I think it's very important to look at what people call these days real-world evidence, patient-reported outcomes, family impressions. And so we're using a number of questionnaires, some of which were developed more generally for patients with pediatric neurodegenerative disorders, and some of which are being in development by us to look at specific aspects of the impact of MLD on the children and the families as part of this study. And taking all those 5 categories together, one hopes that A, they align and agree with each other, and B, that even if one of them separately may not be necessarily, quote, the answer, that taken as a whole, they would indicate or allow a formal assessment of the efficacy of the treatment, or not, as the case may be. And in clinical trials, one always has to say or not. The reason we call these trials is because we don't know whether this is going to work. Everything you've heard presented this morning is a plausible idea, well rooted in good science, mainly animal science originally. But as I mentioned at the beginning, we've already had one experience with MLD of a treatment that was entirely plausible from the animal science. It simply failed in humans. And I don't want to discourage anybody by saying that that could happen again here. We all hope it won't. And I think we've got good reason to believe that all of the things we're talking about can and will succeed. But in a highly regulated industry like the pharmaceutical industry, one has to meet a very, very high bar of benefit to risk for any therapy. And that's how I look at approaching it. Sorry, it's a rather lengthy answer, but maybe it helps get people oriented.
No, I appreciate that. So let me ask a follow-up here, and others can certainly contribute as well. But so MLD and other disorders like GLD are complex because there's demyelination both in the central side and then on the peripheral side. And, you know, I mean, ultimately we want to make people better, but if we're getting more benefit in one of those aspects than others, I mean, how do we help sort those things out? And is nerve conduction studies, for instance, you know, are those important endpoints for these types of things too? I mean, one can envision if you're giving intrathecal drug, you might get a better response centrally than you might peripherally, for instance, and being able to sort out the relative benefits might be, you know, key to figuring out what next steps should be.
Yeah, that's a good point. And that's actually the reason why we chose, and Albert, who was working with us at the time, will remember this, that we chose to go through the intrathecal route. We wanted to address both the peripheral and central nerves. Yes, there are a variety of techniques that can be used to assess peripheral nerve function. The primarily ones are electrophysiological, the EMG and nerve conduction velocities. Older techniques for doing that, as some people may be aware, can be quite painful and difficult for the subject. However, there are more modern techniques available now, particularly developed by people at the Mayo, that would allow one to look at nerve conduction velocities in a more benign way. This is about to be published, but I'm happy to share it. We actually did study peripheral nerve function, including biopsies, in the phase 1 trial that we did. And we have anatomical evidence that will be published shortly that indicates some recovery as judged by volume ratios of the myelin to the rest of the nerve to put a simple example, and some others indicating some response to the peripheral administration of the enzyme. And, you know, from a clinician's perspective, which is what I always have been and will be, it's hard sometimes to sort out the peripheral and central components of this disease. Anatomically, you can easily sort them out. But if you're looking at a particular child and saying, is this child not walking because it's a central effect or a peripheral effect, it can often be difficult to make that judgment. But it was encouraging to us to see those changes in peripheral nerve, and we've seen them in both animals and in some of the patients from the phase 1 study who underwent biopsies.
That's really interesting, David. One of the things that we're also interested in is, are there things that we can measure preclinically that we think would translate into the clinic? And I think that's something that's big. As you had mentioned, some of the early data looked really good in the preclinical models. And I think it's something that I think the whole community faces. How can we improve that translation? And are there things that we can really study preclinically in appropriate models that may help us generate that benefit risk determination prior to even going into the clinic? I don't know if you have thoughts on that.
No, absolutely. But I think that's one of the reasons why one of the reasons why one has to have so many different functional perspectives involved in developing a research program. And you and I have worked together before, so my comments aren't going to surprise you, but one really needs to have people from every possible background, which in my mind includes, where possible, the patients and certainly their parents, in designing a research program of this kind right from the beginning. And as you know, we tend to have clinical people on our early research teams. We also have scientists like yourself on our late clinical development teams for exactly that reason. I imagine that all the other companies are doing the same thing now. And that's certainly evolved over my time in industry. When I first came into industry, things were a lot more siloed. And but one needs to look at it from every possible angle and develop collectively an assessment of how are we going to show that this either works or unfortunately doesn't work, but at least we get a clear answer.
Right.
Well, in terms of these biochemical markers or imaging markers, for instance, I mean, one of the things is how to use those in terms of outcomes, related to your disorders. But another might be a predictive value of those things in terms of prognosis and how people may respond to therapy. And so I mean, maybe people want to expand on that a bit. And related to that, so we've been interested in neuroinflammation, and what's the role of inflammation actually in MLD? And is that something we should be monitoring? Is that potentially meaningful?
I think it is, and I didn't specify those, but we are in fact looking at various inflammatory markers as well. And I imagine, and perhaps Bobby or Albert might want to speak to this, I imagine they are or are planning to as well. Clearly this is an inflammatory process, whether one looks at it at the level of inflammatory molecules or whether one just looks at it at the level of the MRI. And one of the questions that we've had to face, and I know Albert and I have discussed this before, but I'm sure everybody else has come to it as well, is what is the optimal timing for treatment of this disease? Now, from first principles, you could say as soon as possible when diagnosed, and nobody's going to disagree with that. However, how far does the disease progress before any therapy can no longer control it? And this, in my mind, links to how brain growth occurs in children. We're dealing with this particular process, particularly of myelination here, where it begins in late pregnancy, it progresses, but there are spurts of brain growth, particularly as the child starts to walk and so forth. And we were faced with the question of dose, because it was clear from our earlier study that whilst there was some signal there, we were not at the time giving the children enough enzyme. I'm not sure that what we're giving right now is necessarily the right dose. It's what we call the maximum feasible dose. It's what we could get in through the device on a reasonable schedule within the safety limits, because you have to focus on the safety limits as well. But we're still exploring that. And I think what happens with these children is that at a certain point in their development, this enzyme is maybe less important. And then as myelination develops, sulfatides are part of the normal component of myelin. So like many other things in biology, a certain amount is a good thing, but too much is toxic. But then you get into these spurts in brain growth where the production of either of sulfatide and probably secondary peptides becomes overwhelming and leads to inflammation and leads to, and to the damage.
Great. Well, I wanted to talk a little bit about the importance of patient selection for some of these interventions too, and maybe I'll take a step back and talk a little bit about transplantation, because that's where we probably have most of the data in terms of intervening for MLD, and the transplant studies really started in the mid 80s, really, and the concept there was based on cross correction, which Liz Neufeld's group and others back in the late 60s essentially showed that if you had cells from a patient that had a lysosomal disorder and you put them in tissue culture with normal cells, that the normal cells could correct the accumulation of whatever substrate it was. And that's really the foundation, I think, for moving ahead towards transplant. So the idea was that if you could transplant normal cells making normal amounts of enzyme into a patient that had a lysosomal disorder like MLD, those blood cells would circulate through the body and provide enzyme at least at normal levels, and that enzyme produced would be able to correct the cells that are deficient in enzymes. So that really was the impetus to get started. And like I say, there's a substantial amount of experience now in transplant for MLD. Although it's become very clear that it's it's easier to help prevent disease than it is to make somebody better if they've already gotten sick. So that's very clear. And then within the various phenotypes, for instance, late infantile versus juvenile versus late juvenile or adults, the rate of progression ends up being important if one is trying to intervene. And then within those groups, the earlier you can catch the disease and intervene, the better opportunity you have to make someone get the optimal outcome. So, in terms of that, maybe I'll ask Bobby, and then Seymour can fill in as well. But so what do we know about using these interventions for patients with various phenotypes, and what their clinical status is prior to intervention, and how that affects outcomes?
So maybe I think the first point is kind of a more general point that is really kind of related to the preclinical data on enzyme expression, Paul, that maybe I could address first of all. And clearly, we're using an autologous HSC gene therapy approach as an alternative, perhaps, to a transplant approach. And we know that with a transplant approach, there are still kind of deficits that patients have, or there are incomplete reconstitution or incomplete correction of the disease phenotype. So, and while transplant with wild type donor cells provides a certain level of enzyme expression that can be taken up by target tissues, the question is really, does it provide enough to allow full correction to occur? And that's where I think this concept of an HSC gene therapy, an autologous HSC gene therapy, where you kind of overexpress the enzyme through genetic engineering, has a potential effect. And so, we've studied murine models in both MLD, in MPS 1, MPS 3A, where there's kind of parallels in all of these, suggests that if you do a wild type procedure, you get some correction, but not full correction of the disease phenotype. And it's the overexpression of enzyme through kind of genetic engineering of the autologous cells that seem to provide the best outcomes. And that was seen consistently in the MLD, MPS 1 and the MPS 3A studies. And so you can do that through, the use of promoters that express the transgene effectively, through introducing multiple copies of the gene into HSC. So that allows an overexpression to occur. So I think that's the principle on which we've taken forward the MLD program, and now the MPS 1 and MPS 3A as well, that overexpression and providing enzyme in the CNS that can be taken up by neurons to prevent the neurodegeneration. So I think that's the first thing, is the level of enzyme expression in the right space is important to correct the underlying disorder. And then to your second point of what patients to treat, I think, and this is almost universal in these very severe neurological diseases, is the earlier the better. And I think that's absolutely clear. Whatever intervention that you're using is, you want to go as early as possible and before neurodegeneration has started to take hold. And certainly in the clinical trials that we've conducted with the HSC gene therapy for MLD, we've seen the best outcomes in the either pre-symptomatic patients or those at the very early stages of neurological deterioration. And so I think so that's why early diagnosis, disease awareness, newborn screening, which I know the Cure MLD Foundation and others are very, very supportive of, is so important in this disease. And that early diagnosis screening has to be coupled with intervention at an early stage.
Yeah, maybe I can just speak to that a little bit as well, because it came up in the planning for our trials. At one level, MLD is, of course, biochemically all one disease, but clinically there's a very broad spectrum. The oldest patient I ever diagnosed with MLD was 67 years old of age. Perhaps an unusual situation, but nonetheless, it happened. Somebody who actually had been arrested for drunk driving and could not be found to have any alcohol in his bloodstream. So that's a story for another time. But in the discussions that we've had with the regulatory agencies around the world as we were putting the most recent trial together, this question came up. And there are several considerations. Obviously, one wants to be able to treat, particularly with something that one thinks is going to work, as broad a range of patients as possible, as soon as possible. However, from the point of view of the regulators, and we do live in a very highly regulated industry, they are looking for a demonstration generally in the most homogeneous group of patients at the beginning. There's a lot of variation between individuals. I don't need to tell anybody on this call this. Everybody with MLD is different in good ways as well as not so good ways. And unfortunately, when you're doing a clinical trial, that variation can interfere with detecting what we formally call a signal, the response to a therapy. So generally, one is pushed by regulators to study the most homogeneous group, and typically the one with the best available natural history. And in this case, and in yours as well, that's come down to the patients with the earlier forms of late infantile MLD. But I do get asked regularly, and I'm sure you do as well, what are we going to do about juvenile patients? What are we going to do about adult patients? The answer essentially has to be, I think, and I'm open to other opinions on this obviously, is that one has to demonstrate proof of concept in the most homogeneous and most rapidly changing group first. But then one expands, and I see you doing this at Orchard already, to look at other groups, particularly in the earliest stages of their disease.
Well, let me take a bit of a step back too and talk about transplantation and that process. Then I like kind of contrast it a bit to both the ex vivo and in vivo opportunities for therapy. So, just as background, again, starting back in the 80s, we started using chemotherapy protocols that were derived for patients that had leukemia primarily, and utilize those for patients that had metabolic disorders. And the process essentially is to identify a donor who hopefully is making normal amounts of enzyme. And for instance, a brother or sister might serve as the donor, but if they're carriers, then that may be problematic if we're thinking that the amount of enzyme is important in achieving the best outcome we can have. But essentially, The procedures define the donor. We treat patients with chemotherapy to eliminate virtually all their blood-producing cells, but also to knock out their immune system so they can accept cells from another donor. And there's a number of different complications that can occur as part of that. There's the chemotherapy-related ones, but there's also the immunologic ones, as people can reject the donor cells, or the donor cells can attempt to reject the new body that they're in, which essentially is graft versus host disease. So there's a lot of, we've made, I'd like to think, a lot of progress in terms of allogeneic transplantation and the ability to find good donors and supportive care and whatnot. But there's clearly still significant risks associated with allogeneic transplant. And so, you know, when I think about comparing opportunities for therapy, there's really, you know, maybe two major ways of looking at this. One is the risks associated with the therapy, and the other is the efficacy, or how much better you can make the patient than you can otherwise have achieved previously. So, you know, again, allogeneic transplant is safer than it used to be, but still we clearly lose patients going through the transplant process, and there's long-term complications associated with that. So the autologous approach, maybe we'll start there with the lenti-based therapies. So the autologous approach, which still utilizes chemotherapy, but maybe not as intensive chemotherapy as we'd otherwise use, but chemotherapy is still required. And you know, Dr. Gaspar, I mean, we kind of ask you a little bit about where that piece is going. But and then on the efficacy side, if we think that the amount of enzyme delivered is important in alleviating the disease, you have the opportunity, as you mentioned, to get more expression in using gene therapy. So it has the potential to be not only safer, so because it eliminates some of the complications using somebody else's cells, but has the opportunity to be more effective as well. So that was a long preamble for a question, but Bobby, maybe you can expand on that.
Well, yes, thank you, Paul. And you know, from my previous life, we've kind of talked about conditioning regimes and transplantation, etc. And I think you're right. You know, the use of autologous cells just kind of firstly takes away that major element of transplantation that we all kind of try and cope with, which is graft versus host disease. So by using autologous cells, we avoid that particular risk, and that can make an autologous HSC gene therapy procedure safer. As far as the conditioning is concerned, currently an HSC gene therapy procedure still uses conditioning to make space for the incoming cells. And when you contrast with what has to be done with an allogeneic setting, perhaps we use slightly less in terms of chemotherapy than using the allo setting. So there's a potential safety element there as well in the autologous setting. We still use, for MLD gene therapy, we use busulfan. And I think it's an important point to note that the busulfan is used for two purposes, really. One is for the incoming gene modified cells to make space for them in the bone marrow. But also it is clear that one of the ways this may potentially work is through the ablation of these microglial cells in the CNS. So that's the other thing that busulfan does, is to deplete resident microglial cells and therefore make space for incoming gene modified cells to fill that niche. And there have been preclinical studies that have been done which demonstrate that probably busulfan, better than irradiation or other agents, has that property to deplete or better deplete those microglial cells. So at the moment, the potential benefits that we're seeing through HSC gene therapy in MLD does require this kind of busulfan-based conditioning to make space for the gene-modified cells to go to the CNS. So I think that is, you know, and then you talked about the overexpression, which I alluded to previously as well. So you make space, the cells come into the CNS, and then they're overexpressing enzyme, and those are the advantages in terms of both safety and efficacy for potential correction. I would say that, you know, as the field moves further on, you know, I think the way we condition patients is also going to improve. And, you know, you and I have seen how conditioning has evolved over time. We've got better kind of therapeutic dose monitoring of busulfan to keep it within a specific therapeutic window. There are other agents available. We're seeing the advent of antibody-based conditioning. But I think, you know, there's also potential for using other agents. You know, I mean, they're experimental at the moment, but perhaps in the future, we'll get into the clinic where you can use a specific targeted molecules to even deplete microglia. So we don't have to go down you know, a systemic chemotherapy route for this kind of approach. So I think, you know, currently using HSC gene therapy, there is a need to deplete, and I say busulfan is what we're doing now, but maybe there'll be other ways of doing it in the future.
Can you help us better understand? So if we're going to modify the preparative, so if you have a retrovirus or a lentivirus that you're happy with, and it looks like you're getting the expression and the transduction and whatnot that you need, but if you want to modify the preparative regimen and potentially make it safer, how is that going to be done on a licensing sort of basis? I mean, do you have enough flexibility when you get a license to use an approach that you can modify those types of things, or how are the regulatory groups going to envision the modifications of the preparative regimen?
Yeah, no, it's a really interesting question, because the conditioning regime is not part of the label. So it is not part of the label. So it's essentially a recommendation. So, you know, in the SMPC document, you basically, the product are the gene modified cells. And what we do is we state that in clinical studies, this has been used with a busulfan based conditioning regime. And so there's no obligation to use busulfan in clinical practice. It's a recommendation, or, you know, a kind of guidance from the developer. So in the future, if there are alternative regimes available, there is you know, discretion to use alternative regimes, because you know, the product is not attached to a specific conditioning regime. It's clearly what experience has been, what the experience has been in the development.
Hmm. Well, and things clearly are different in Europe than in the US. But so if you modify things significantly, are the insurance providers going to look at those things differently and basically say this is not what the how things were developed, and so, you know, we're not going to pay for it if you deviate from where the data really is?
Sorry, David.
No, I was going to say I certainly see that, not particularly for this disease, but others. I mean, I think one of the important things, I think, for everybody to understand here is that over the last 20, 25 years, the regulatory landscape has changed. When I started working in this field, it was easy to get a label to cover all of the cases of a particular disease: Fabry-Gaucher, Hunter syndrome, Hurler syndrome. All those labels simply state for the treatment of Hunter syndrome, for example. No regulatory agency does that anymore. They base the label largely on the clinical trial protocol that they're approving, and particularly the US FDA, they're the most aggressively restrictive about that. I think your case in terms of conditioning is separate, because the conditioning routines are kind of part of standard medical practice anyway. But certainly in terms of, say, my enzyme treatment for MLD, the label initially will be restricted to the group we studied. And somebody was asking online again about access for juvenile and adult patients. And in the end, additional studies will have to be done to provide access to those patients, whereas 20, 30 years ago, that would probably not have been necessary.
So, Bobby, I want to move to Dr. Seymour here in a second and kind of ask more about the AV kind of approaches. But before we do that, so have any adults been treated with lentiviral approaches yet? I mean, is there flexibility to potentially do that, or is the label fairly specific in terms of which populations one can go after?
So Paul, so the label in the approved product in the EU is specifically for patients with the late infantile and the early symptomatic, early
Right.
form of MLD. So it is restricted to that population.
So if we wanted to potentially expand that into, the adult population, for instance, one would have to do that as a separate trial, presumptively.
I think that's the way we'd have to look at it, as to whether we do a specific trial for the late juvenile adult population. I mean, I think there has been talk about whether you use real world data with patients treated on a compassionate use basis and take that to the regulators. But I think in the first instance, it would have to be done through some form of formal trial of late juvenile and adult patients.
Yeah, and to just come back to the access and reimbursement issue, you're right. I mean, in Europe, you're very much more tied to the actual nature of the study that you did. In the United States, to some extent, first of all, there's more prescribing flexibility for the physicians because of the allowance of off-label use. And secondly, particularly for rare diseases, some of the insurers will look at a rare disease in a more tolerant way, I should say, perhaps. Still difficult, still requiring a lot of administrative burden and paperwork on the family and the physicians, but one can to some extent drive it in the US, whereas in Europe you tend to be locked into what's in the SMPC.
Okay, so Dr. Seymour, we were talking a little bit about the autologous approach with lentiviral, vectors, for instance, and having to get chemotherapy up front, and then the patients are going to get neutropenic, and we'll have all the supportive care that's part and parcel of that. So I mean, that doesn't exist for the in vivo AAV types of approaches, but there are concerns about immunologic aspects towards AAV, and so there are patients that may not be eligible to get AAV therapy, kind of based on what their immunologic status kinds of looks like. And then there's also the piece about being on immune suppression, at least for some time after the vector is delivered. Can you walk us through some of that a bit?
Yeah, absolutely. And maybe what I'll refer to here is where we have clinical experience with our AAV approach. It's in a disease called PKU. It's adult PKU. What we had to develop there, because AAVs are naturally occurring, so a lot of people in the population have been exposed to them in a variety of different serotypes. And so what we have developed is a screening assay so that we can assess prior to getting the therapy whether or not a patient has titers, so neutralizing any kind of antibody that would neutralize the uptake of the AAV to the cells. Because if that happens, then it'll prevent the AAV from delivering the vector genome to the appropriate place. For ours in particular, it's about 80% of the population seems to be amenable with really no neutralizing that we can detect. About 20% do have some titers, and we published a paper on that a couple of years ago to show that. So as we think about going into a trial, and to Dr. Whiteman's assessment, we want to make that trial as homogeneous as possible. We're very conservative on the neutralizing activity of that. So we want patients to enroll that really have no pre-existing neutralizing activity, so that they can get in. Interestingly, we haven't had a screen fail to date into the trial, but we certainly have to do that. The second part that you had referred to is around the steroid regimen, because we are administering an AAV that is a foreign capsid that's a viral particle, and so the body will mount an immune response to that. And it can be both a humoral, meaning that they'll generate antibodies against that capsid, as well as a T cell mediated approach. So what we've done, and I think the field in AAV, in vivo and AAV, has been doing for a while now, is putting patients on the prednisolone and steroid regimens prophylactically. So they'll start a tapering of that a day before dose, and then you follow them over time. Really measuring any kind of liver enzymes like ALT, AST to evaluate that. So that's how we're approaching that. The other big part in the field is around readministration. So as you do a one-time administration, all of the patients that will get that will mount some immune response humorally and generating antibodies to that. a lot of the community now is looking, if you go 5, 10, 15 years down the road, could you re-administer if the expression starts to wane a little bit? And a lot of people are looking at that. Either can you come in with a different viral serotype that doesn't have the epitope of the one you used, or can you suppress the immune system long enough to then have the transduction happen so you can get around the antibody-mediated aspect.
Great, that's helpful. So, so with the current trials, how long is the immune suppression piece? How long is someone on prednisone, for instance?
So what we've been doing is we've been trying to do that through periods where historically you've seen some transaminases. So the regimens can range anywhere from maybe 8 weeks up to through 20 weeks, depending on the AAV. Now you're not on high dose prednisolone the whole time on 20 weeks. You will be going through the taper over that time period, but you typically start at a higher dose, like around 60 milligrams a day.
Okay. We've been talking a bit about the regulatory sides of things, and I actually kind of wanted to get into a bit because I think people listening may be interested in the insurance provider side of things. So OTL-200 or the MLD lentiviral vector is now approved in Europe. So what are the implications for the various countries in Europe? I mean, are they mandated to support patients being treated on this now, or is that a country by country designation, or how does that work?
Yeah, it's a country by country negotiation. So we go through discussions with reimbursements agencies in the various different member states, the UK, France, Italy, Germany, for example. So we're going through those negotiations now.
Okay, and maybe Drew or Dr. Seymour, Dr. Whiteman, I mean, if you guys want to weigh in on what you think the landscape is going to be like in the US too, as some of these therapies become available, because I'm sure those discussions are ongoing, because there's an awful lot of insurance providers in the US, and there's private insurance and state insurance, and so there'll be a lot of things to navigate, because presumptively some of these therapies are going to be quite pricey. So does anybody want to take a shot at what the landscape may look like there?
I can give you a historical perspective, not specifically for these therapies, but for things that I've worked with over the years. I mean, first of all, as in Europe, the insurers will primarily look to the label to guide them, but as I mentioned earlier, there's more flexibility here. What we found, and I think others have as well, is that the large insurers that often have their own rare disease medical teams and rare disease medical directors tend to look at this rather pragmatically, and although these therapies are expensive, they realize very quickly that their total exposure in an insurance sense is small, because the number of patients is small even for a large insurer. Most of the difficulties we have are with government programs and with smaller insurers where their risk pool is smaller and their resources are less. But I don't want to name names, but there's one large insurer that you can easily approach their medical department and fairly quickly convince them of the utility of a therapy, if in fact you've got good evidence for it, in a way that they won't challenge. There are others that give you a harder time. It has to be individualized. And in Europe, it's very much country by country. First of all, in France and Italy, you can get earlier access because of legislation there. In other countries, Britain is the most notorious one. It takes even longer. Sorry. Go ahead, Albert. I'm sorry.
I was just going to add an additional comment on it. And I think it's one of the things that's so important as you're through the development of these products to engage with the payers very early. So bring them along and really start meeting with them as early as you can, laying out the target product profile, and then obviously with your evidence base that you have from your trials seems to help as well. And it's a fairly large effort. You don't want to get to the end and then surprise the payers. You really want to bring them along as you're going.
Yeah, absolutely, and I think all of us are doing that all the time. It's one of a number of things we have to do to move this forward.
Yeah, and again, as we're kind of as if following the approval of the EU, and as we're talking to the regulators in the US, we're also doing the same thing with the with the insurance agencies. It's really about the nature of the disease. I mean, it's a very rare disease. Most people won't have heard about it. So to understand exactly what impact this has upon the patients and what potential benefits these treatments can bring. So to kind of talk about the disease, educate about the disease and about the potential of the therapy, so that you've already had those conversations, as you all said, before it's actually approved.
Related to this, what can some of the advocacy groups like Cure MLD do to help move some of these things forward, or even individual, families or those that are potentially interested in having these opportunities? Are there things that they can do to help, decrease the barriers that may exist for implementing some of these studies and some of the therapies?
I mean, I think the first thing is, I would say thank you very much to the patient advocacy groups like Cure MLD for all that they're doing to raise awareness of the disease even now, and and the work that they're doing with policymakers, with regulators, to highlight what the devastation that this disease can bring. So I think that's the kind of educational activity coming from the families and the patients themselves that can really raise awareness, so that when there are therapies that are coming along and being approved, that information is already available, that background is known by the various different stakeholders. So again, it's so much more powerful when it comes from the patient support groups, far more powerful than any of us saying, this is what the disease does and this is effects, because if they see it firsthand, then they understand why there is a need to have, to develop therapies, why there is a need to have these therapies reimbursed appropriately, so that there is access. And there's no more powerful voice, in my opinion.
Yeah, I completely agree. And I think the other thing too is really hearing firsthand, we talked a little bit about endpoints. What are some of the things that we can evaluate as part of the trials, really understanding what are patients and caregivers of those patients really facing on a day-to-day basis, that we can start to build into the trials to help from a development perspective of that as well. So.
And then treating physicians are also part of the advocacy in terms of securing access to treatment. I mean, this has to be a community effort, and at another level, and sort of led by the Every Life Foundation and groups like that, this is being tackled sort of more of a national, international level. But it's a constant push to secure funds to drive this kind of work, and everybody has to join in, and they do.
And I think it is difficult, because when if you're talking about the regulatory agencies or the reimbursement agencies, etc., they have so many things that they're looking at, so many diseases that they're looking at, and we're kind of immersed in one particular disease where we kind of know what it's about and everything else, and our whole world is about it. Whereas they're looking at hundreds of different and so the more powerful the voice is, patient support groups and KOLs talking about this firsthand, and to leave an impression on their mind what this disease is, makes a big difference, a really big difference.
The regulatory agencies are much more open now. I mean, there are formal mechanisms for family and advocacy groups to meet with them, and in the same kind of way that we as companies can meet with them periodically, and everybody should be taking advantage of that.
So one could envision, I mean, with Europe being primarily national healthcare based, it's, maybe that approaches are the same throughout, Germany or France or the UK, for instance. But in the US, obviously, things are a lot more complex. And, if there are large insurance groups that are private groups that people are getting their insurance through, that may give you a fair amount of flexibility in terms of where you might go for some of these therapies and what you might be able to offer. But enzyme therapy may be a bit different, because if there's an approved enzyme, that could potentially get distributed to all the pharmacies. But if if there's cellular therapy that's quite specialized and there's going, or gene therapy, and it's going to be limited, at least to start, to several different centers, then presumptively people are going to have to travel to get these kinds of interventions performed. And if they live in a state where, for instance, there isn't a center performing this, they may have to leave and do that. And we certainly on the bone marrow transplant side had to deal with these things on an ongoing basis. If somebody has, for instance, state insurance that doesn't reimburse very well, our hospital won't let us bring a patient in from across the country if we're going to lose money. So, how are these things going to get dealt with? And Drew, maybe you want to I know you guys have been working on this as well. So how are those things going to be impacted down the road? I mean, do we need to have national policy for these types of interventions especially for these rare disorders, or what's the way forward?
Yeah, it's a really great question, Paul, and definitely agree with everything that's already been said about the complexities here. And then you start to layer on top of that the difference in the United States between private insurance and public insurance, and those pieces. And the point made earlier about getting out ahead and really talking to both private insurers and public insurers and starting to understand not only about the disease itself, but sort of about some of the technical aspects of therapy delivery and some of those pieces. And it's ultimately in the long run going to be a discussion that does involve the patient organizations, the healthcare providers, and the companies in conjunction with all of those insurers to sort of find these pathways. But in each scenario, as you're saying, like, for example, crossing state lines and some of those kind of things, there's sort of often sort of needs to be even a unique situation worked out.
Yeah, I mean, certainly with the kind of the HSC gene therapies, it's a kind of, it's a paradigm shift in treatment, isn't it? I mean, it's not a standard conventional therapy. And just like we're, kind of challenging, things on a regulatory front as we get these things approved, we'll be challenging on the reimbursement front as well, and, setting up new models of payment. I do think there is there's learning to be had, Paul, from the kind of bone marrow transplantation field, where you do have specialist centers that can offer transplantation for specific rare diseases. And I think, the model that, we're certainly working towards in Europe and the US will be, qualified treatment centers, so area, centers of specialty that will be able to administer the therapy. And so we'll have to work towards how we coordinate with these qualified treatment centers, with the payers, to be able to offer that therapy to as many patients as possible, if not all patients. So it is, again, a process of coordinating with the various different stakeholders, educating the reimbursement agencies, and getting to a point where we have mechanisms of being able to offer this to all patients who may need it. But it's going to be a different model because this is different to other therapies that have existed previously, and we're just challenging, let's say, conventional paradigms the whole time.
Yeah, that's such a great point. Absolutely.
Well, we've got a number of questions from the audience, and I want to move ahead towards that. But maybe before we do that, just to make sure the audience kind of understands where we are with emerging therapies here, maybe if I could just have Dr. Gaspar, Dr. Seymour, Dr. Whiteman, and Drew, if you wanted to as well, just talk about what the status is in terms of your current intervention. I mean, who may be eligible, what the timelines look like, maybe if you can do that in like 2 minutes or something, that would be great. So Dr. Gaspar, maybe we'll start with you.
Yes, so lentiviral ex vivo autologous HSC gene therapy has been approved in the EU by the EMA. The label is for pre-symptomatic patients, late infantile, and also early symptomatic, early juvenile patients. So that is what it's indicated for. I say it was approved at the end of last year. And we're, as I say, working with the reimbursement agencies in EU countries to be able to make that available, and also through early access routes as well in countries where that legislation is available. We're working with the FDA to try and bring that to the US, and so we're in active engagement with the FDA to determine the path to filing. And once we have clarity on that, then we could talk more about what that timeline would look like.
Okay, Dr. Seymour.
Yep. So for ours, AAV-mediated gene therapy, as you saw from the data that we had presented, we're still in the preclinical realms. We're in what we call IND-enabling studies, where we're really evaluating its, from a safety as well as a manufacturing, and then from a clinical trial design, we are anticipating that we would initiate in the late infantile population. I think for many of the reasons that were discussed earlier about the natural history, assessing whether or not the therapy, is having an impact sooner. We would most likely target the early symptomatic or the symptomatic late infantile population. At least that's where we are now. More will come perhaps with feedback from the FDA as we progress along those realms. But that's kind of, in a sense, where we are with our program as well.
Okay, great. And Dr. Whiteman, where are we with intrathecal enzyme therapy?
Well, essentially, we're fully enrolled at the end of this phase 2b trial. We won't have a readout of data for another 2 years, and that what we do next depends on what that data looks like. Frankly, if it's good enough and the results are clear enough, then obviously we'll try and register the drug immediately, and under the rare disease regulations. If in fact we've only shown that we've amplified the signal, which is a very weak signal really from a regulatory point of view, that we saw in the phase 1 study, then we will likely have to do another study. I don't think one could say much more than that at this stage. Okay,
great. Thanks very much. So I think we'll move on to some of the questions from the audience here. So there's a question about, so Dr. Seymour mentioned the role of mRNA in AAV vectors and whatnot, and there's a question regarding the COVID-related vaccines that also target mRNA, and whether there may be any interactions or difficulties associated with those things.
Yeah, it's a great question. So I mentioned mRNA because that's actually, the AAV will deliver the vector genome, which is DNA, into the cell's nucleus. It will then make the mRNA of aryl sulfatase. So any kind of correlation between that and the COVID vaccine, there really isn't any. The only other thing that I would say though, and we're looking at this, and Dr. Orchard, you brought it up, the steroid regimen where we do preconditioning. If there was going to be a COVID vaccine, we would probably have that vaccination done prior to going on any kind of immune regimen before dosing. This is how we're thinking about it also for our programs that are in the clinic. Because then you may suppress the immune system and limit the amount of immune response to the vaccine.
Okay, great. There was another question. I think we addressed at least some of this in terms of pre-treatment for the homology AAV in vivo vectors, and I think you also kind of touched a bit on the late infant, maybe the early late infantile symptomatic patients would really be what you'd be looking at in terms of the trial, but it's still a bit early to say.
Yeah, exactly. And I think in terms of pre-treatment, there's no pre-treatment for receiving the AAV outside of the steroid regimen that we have part of our trials.
Okay. So as we mentioned earlier, there's certainly interest in patients that have later onset disease, and and related to that, the limitations of blood marrow transplantation. So how would we transition, for instance, into the adult population of patients? I would think, enzyme therapy would be relatively straightforward, but if the label, for instance, for gene therapy is for younger patients, again, would we have to do another trial to be able to offer therapy for the patients that are older? Bobby, can you just kind of weigh in briefly? I know we talked about this a little bit, but
yeah, sorry. I'll just repeat my answer previously, which is, the label, as you say, is for the late infantile and the early symptomatic, early juvenile population. So I think the extension will be dependent on the jurisdiction, as it were, and is what is allowable, whether there is an expansion through compassionate use or expanded access programs, or whether there is the need for a formal trial in those populations.
Yeah, one of the things that is beneficial potentially, if that's the right word, for people that have later onset disease, is that it's progressing on a slower basis. But if one's going to show that you've bent the curve, then it makes it difficult too, because the natural history is that they're going to progress less quickly as well. And so, designing a trial where you can have a meaningful answer within a few years of when you intervene could be difficult as well.
Exactly.
And I think that is one of the challenges, especially in a very rare disease such as this as well, to collect enough patients and to follow them over a long enough period of time in order to see a clinically meaningful outcome could be difficult, because of the natural history of an adult patient is that you start to only see you see deterioration over many years. How do you design a study to be able to do that, unless you have obviously a very good biomarker that correlates with those kind of clinical outcomes?
And a good biomarker is really the only way you're going to be able to get around that. I mean, that's why we designed this present enzyme replacement trial as a 2-year study. We felt that in the first-year study, whilst we had a weak signal, we didn't know that we'd fully evaluated the extent to which the therapy might intervene.
Yeah.
So, and when you're working with older patients, the natural history is less well defined. It always surprises people to hear this, but if you look at MLD in the context of many other rare diseases, we have one of the best defined natural histories because of the work of people in Tübingen in Germany and their successors. And we have some of the best natural history databases because of several people's efforts of any rare disease I've ever had anything to do with. And even then, it's not always that straightforward with the regulators, because the other issue we all face is the issue of controls, and nobody is comfortable using a randomization in this situation.
So let me just ask about that too, because I think the natural history data ends up being, critically important in terms of assessing whether these interventions are helping. So as we move to newborn screening, again, this is going to be, years down the road, presumptively, hopefully sooner rather than later. But so, babies are going to get picked up at birth, and we'll be able to do their genotypes and figure out where the mutations are. But so, we may know what the natural history of late infantile disease or early juvenile disease is, or later onset disease is. But if you're picking up a baby at birth, how tight is the correlation going to be with the genotype, meaning what mutations are you seeing with what you're going to potentially expect those patients to see? I mean, is there a database or a website or something that one can refer to and basically say, okay, I've got this combination of mutations, and based on this, we can predict that this baby would, otherwise get symptomatic by at a year of age, or it would be 10 before you'd expect to see symptoms? I mean, how much data do we have to be able to sort that out?
I think the short answer is not enough. I mean, databases are being built. All of the natural history databases are accumulating this data. Companies are accumulating this data. There are national repositories like the HGMD in Cardiff in the UK. If others want to comment, please go ahead. But I've looked at all of those, and frankly, the situation is somewhat better for MLD than it is for some diseases. There are certain mutations where if you see that a child has 2 copies of them, then you know pretty much that it will be late infantile MLD. However, and conversely, if there's 1 copy of one of those mutations and one of another, then it's usually juvenile. But you don't know where within the late infantile and the juvenile spectrum that particular child will lie. And this is the whole issue of genotype-phenotype correlation that's being talked about a lot in many diseases at the moment. Frankly, it's just not good enough yet. And with the single exception I mentioned, which involves the common splice site mutation that some of you may have heard of, then we don't have good prediction of outcomes from genotype in this disease. And I noticed that Dr. Gelb is one of the people online. I don't think he's able to speak, so I'll ask him to forgive me for speaking for him. But in developing the newborn screening substrates that we now have, in the end, this disease has to be defined biochemically. It's defined as a deficiency of the enzyme with excess sulfatide production and a consistent clinical phenotype. And most people would add with an appropriate MRI, although you have to be careful because in the early stages of the disease, the MRI may actually look normal. You can't define the disease on DNA mutations. I wish you could. All of our lives would be a lot simpler if we could, but the knowledge base to do that doesn't exist. And frankly, I don't think it's going to exist for another 5 to 10 years. I don't know. Albert's a geneticist as well, so I don't know if he wants to speak to this, but that's my view of it.
And I suppose to a certain extent, David, the newborn screening that we're talking about does rely on the biochemical abnormalities as the screen initially, and then we define the genetic abnormality. So there's a kind of an element of newborn screening that already says biochemically these patients are abnormal as far as MLD is concerned. And then we have the genetic mutation. And then on the basis of that, is there any predictability of what the clinical progression would be like in that particular individual? And from my understanding anyway, the lower or those with certain mutations that lead to absence of residual enzymatic activity are more likely, although obviously it's never definitive, to have the most severe course of disease.
Okay, there's another important question. Well, there's several, but at least the next one up was about potential risks of cancer related to interventions, like especially the lentiviral-based platforms. And so for those that may not have heard about this, there was several patients that were on a sickle cell trial, and this is part of the reason we have Dr. Dietz here with us today, but there's several patients on a sickle cell trial that developed leukemia following getting gene therapy with a lentiviral vector. And so there's been a lot of interest and discussion clearly about whether these were events that might happen anyway, or whether these limited to sickle cell patients, or the virus that was used for the sickle cell patients, or the process of introducing the virus, and what that might mean for use of lentiviral vectors in other disorders. So Drew, if you want to tackle that.
Yes, sure. Yeah, obviously it's a really complex topic, and in the slightly historical days of gene therapy, ex vivo gene therapy, before lentivirus was used, gamma retroviral vectors were used, and there are differences about how those viral vectors are constructed, and what we know about vectors actually causing leukemia primarily comes from gamma retroviral vectors. To date in lentiviral studies, over 250 patients have been treated across multiple diseases. And there are no clearly confirmed cases of insertional oncogenesis, or where that lentiviral vector is the reason that leukemia occurs. But that doesn't mean that leukemia wouldn't necessarily occur in clinical trials. And it ultimately needs to be sorted out. Is it possible that the vector is in some way related? Is the vector there at all? Is the vector sort of present but not necessarily driving the process? And those are all of the types of investigations that are underway. For example, in the sickle cell program, where a couple of cases of leukemia have been seen. One of those cases from a couple of years ago, there was no vector present in the leukemia cells, and the investigator deemed that the leukemia was most likely related to busulfan conditioning. In more recent events, there's a lot of work underway to try and really understand the details of what's going on. When you take a step back and think about why leukemia could occur, it turns out that there are a lot of things that come into play. The vector is just one element, and the process of manufacturing is one element, but also the patient's underlying disease. So it's very different to study these technologies in, for example, patients with bone marrow failure syndromes who might have a predisposition to cancer anyway.
Yeah.
There's a suggestion that patients with sickle cell anemia might have higher rates of leukemia in the general population than in the general population. So there's something about the background. There's also been cancers that have been reported after exposure to alkylating agents, so the chemotherapy that's used in the transplant process. And so these things are obviously potentially very multifactorial. They deserve a lot of attention, and certainly that attention is front and center right now. You know, kind of making sure we sort out as many details as possible.
Yeah, if I just add, Paul, I mean, obviously this is something that, you know, we look very carefully at, and I mean, we've treated, I think it's over 160 patients now across 6 different diseases, data stretching back over 10 years in the first patients that have been treated, and haven't seen any evidence of this, any kind of evidence of these kind of myelodysplastic syndromes or leukemias in the patients that we've treated. And when we look at a molecular signal as well, we don't see any evidence of persistent clonal proliferation of gene-modified cells. So, and you know, these the vectors that we've used, the lentiviral vectors we used, were specifically designed to not have these kind of effects of turning on growth promoting genes, and that's been modeled very extensively in preclinical studies and clinical studies. So I think, you know, and obviously, you know, Bluebird are undertaking a number of investigations. You know, it may well be the disease background has an increased susceptibility to these kind of events, and I think, you know, it's important to study this as carefully as possible. I think the final point I would make is, you know, it's obviously about the risk-benefit profile for patients as well, and what is currently available for that particular disease, and what is the benefit risk profile of the intervention.
Yes, absolutely.
So, and if I could just, you know, maybe, and you guys can correct me if I'm wrong on this, but between Bluebird's experience, Orchard Therapeutics' experience, has been well over 300 patients that have been treated thus far. And so, you know, there's been a couple of these reports of leukemia, but we don't know how well connected those are to actual, well, to the viruses certainly, but then the chemotherapy is kind of another piece. But in context with allogeneic transplant, which is kind of what we would be moving away from towards these other interventions, you know, ordinarily I would tell a family that the risk of dying with a transplant is in the 10 to 15% ballpark, depending on, you know, what your availability of donors looks like and whatnot too. So again, I don't think the risk of developing leukemia is going to be zero with lentiviral based approaches, but again, it's a relative risk related issue, I think. So we got a few more minutes and there's a number of different questions. So I may try to kind of pull a few of these together. So there was some questions about, you know, conditioning with transplant and actually with the gene therapy approaches and what might be developing over the next decade or so. And Bobby talked about this a bit, but there's some of the things that we've been interested in. One is being able to expand a stem cell population. So for instance, if you can transduce a cell population and now you're expressing the aryl sulfatase gene, and you can expand those cells in tissue culture essentially. So you have 100-fold more cells than you would otherwise have, then maybe you don't need as much chemotherapy to get the same effect. So, I mean, that may be one approach that that people are going to take. Obviously, just being able to decrease the chemotherapy might be one thing that we can do, or combinations of chemotherapy. And, again, Bobby kind of alluded to this as well, but there are antibodies that are being developed that might actually help knock out the blood cells within the bone marrow. And so they will take away a lot of the necessary component of the chemotherapy that required to do those things. So I think all those things are going to get looked at over a period of time. But how quickly those might be implemented is tough to know. And at least for MLD, as opposed to some of the other lysosomal disorders, again, if we really feel like an agent that penetrates into the central nervous system to get rid of the existing microglial cells so they can be replaced by ones that are expressing enzyme, busulfan seems to be one of the best things that we've got at this point. So we may be locked into busulfan at some dose, and I don't know what the optimal dose is, and maybe Bobby could speak to that, but it seems like it's unlikely we're going to totally get away from busulfan in some of the neurologic disorders, at least in the near future. Is that fair to say?
I think that's fair to say, actually. I mean, because busulfan has that effect on depleting most of the microglial cells, I think we are committed to using that for the time being, at least, while we look at other agents that can both allow engraftment in the marrow, but also allow depletion in the CNS. So I think that's, but this field to me seems to evolve very quickly. So things move when there's a need. There is a kind of a rapid progression of development. But for the moment, anyway, I think this is what we'll be using.
Okay. So Maria had a question about whether I think whether some of these interventions may be used in combination down the road. And, for instance, one could anticipate that enzyme therapy might be able to be used in the short term before definitive therapy like gene therapy is available. And, I think we're speculating at this point what that might actually look like. But as these become available and licensed, then there may be opportunities to do those types of things. But it's difficult to use two interventional therapies under an IND, for instance, at the same time.
Very difficult.
So, that's a very tough nut to crack with the FDA. So you almost have to get at least one of those things approved, I would think, before you could ask those questions, as opposed to in combination. So,
yeah, I agree.
Maybe one other question I think would be really interesting to discuss here. So for some of these disorders where there's essentially no enzyme made, so there are null mutations or severe mutations and no enzyme is made, and now you engineer cells to make enzyme, is there an opportunity for the immune system to now see this enzyme that's being produced as a protein that shouldn't be in the body, and so you mount an immune response to that? I mean, could you make antibody to endogenously produce enzyme following gene therapy, and how are we looking at, and what's the landscape there?
Yeah, so in the studies that we've done, that has been looked at in a small minority of patients. Some did develop some anti-ARSA antibodies, but they didn't affect in any way the activity of the ARSA enzyme, and they actually were able to resolve over time. And I think one child needed some intervention, but the others were able to resolve their antibody production spontaneously. So that was quite encouraging, and it might mean that ARSA itself is not a particularly immunogenic enzyme. And David may talk in more detail about that. The longer term is that actually, because you're engrafting hematopoietic stem cells and you're allowing them now and you're now allowing education of the immune system through thymic education, you actually over time develop central tolerance to the new transgene. So there may be an element of immunogenicity in the short term, but over the long term, as regulated T cells develop, then you won't have that kind of effect.
Is there any sense that immune tolerance may be different in the younger patients as opposed to patients that are older?
Potentially, although it's difficult to know from the studies that have been done so far.
Yeah,
Okay.
I just echo what Bobby was saying. The only hard data I have, obviously, is from our phase 1 study, which is finished and written up. But the generation of enzymes there, and in particular, of antibodies there, and in particular of neutralizing antibodies that are shown to be diminishing the effect of the enzyme in the lab and therefore presumably clinically, was very low. In the present study, which is not randomized because it can't be, I and my medical directors are blinded to the data until the end of the study. However, the data for safety purposes is reported to us. In other words, if somebody were to have an extraordinarily high antibody titer or an extraordinarily neutralizing antibody titer, or in association with a reaction, then I would know about it. For what it's worth, we haven't seen that yet. I have a sense, and I don't think the data exists in a research sense to fully confirm this statement, but I have a sense that complete, in genetic sense, null deficiency of ASA is probably an embryonic lethal. In other words, patients who are completely deficient in enzyme may never completely survive pregnancy. I don't know that, I can't prove it, but it's my intuition. And certainly the vast majority of MLD patients that I've been in contact with, either in practice when I was and subsequently in industry, are low antibody generators.
Great. Unfortunately, I think we're out of time. So if I could just really briefly in closing say thanks to all the panelists that were with us today. It was extremely interesting and I think informative. Thanks also to all those that tuned in with us today for joining us. I want to thank the sponsors as well, Takeda, Thermo Fisher, and Orchard, who helped kind of make this possible. And especially I want to thank the Cure MLD group that helped put together this series. So this is the last of the 2021 series for standards of care for MLD. So if people have additional questions either about the therapies, for instance, and want to get on the website and find out what the opportunities are there, certainly feel free to do that. And to follow Cure MLD as well. So again, on behalf of the group, thanks very much for joining us today, and for panelists for all the information provided.
Thank you, Raj. Thank you. Thanks for the invitation.
Thanks, Paul.
Thank you, everybody. Thank you. Bye now.
Thank you.